Well, Christy, I show us at the top of the hour. What do you think? Should we kick it off?
Sounds good. This is wonderful. Thank you very much. Welcome, everyone, to the 2024 MCBK North America chapter meeting. This is an exciting launch of this initiative focused on North America, and we're really excited about the program. So I'm going to turn it over to Josh for some comments.
Thanks, Christy. And I just want to say how pleased I am that we've gotten to this exciting two-day event. It's happened through a lot of effort and planning and hard work by a team of folks that have been working on this for some time now, I think, to bring together a really exciting program of speakers, posters, demonstrations, and updates about what each of our work groups have been up to and what they're looking to achieve in the coming year. I would like to specifically thank members of the program committee who have been instrumental in helping us shape this chapter meeting that we will be all participating in for today and tomorrow. I'd like to thank our presenters who have really exciting discussions and topics to cover, as well as our poster presenters and demonstration presenters, and all who have had a hand in shaping this MCBK North America chapter meeting. I'd particularly like to point out and thank the extensive efforts by Noor Khan, who has been there through and through in helping to manage and coordinate and collaborate throughout this entire process. Noor, thank you so much. As well as Janelle Burley, who has been fantastic and instrumental in helping to coordinate and making sure all the technical pieces work together. Before we go too much further into the program, I would also like to extend my thanks to Chuck Friedman, who has shown his leadership and provided guidance throughout this process of bringing together this exciting program and first annual chapter meeting, as well as all the efforts, Chuck, that you have endeavored upon in leading MCBK over the years. Would you like to offer us some comments and observations before we get too much further underway?
Sure. Thanks so much, Josh and Christy. It's really a pleasure for me to be here, and I just want to begin by thanking Josh and Christy for their leadership as co-program chairs of this meeting, which is going to be a wonderful experience for everyone. I'm also very much aware that we are in a transition. As many of you know, MCBK, which began in 2018 as a singular, largely North American movement, has, particularly because of the rapid development of MCBK in the UK, become at least a multinational and maybe even global entity. So we have moved to having continental national chapters of MCBK, and as a result of that, we are having the first meeting of the North America chapter of MCBK today. And with that is going to come other changes in governance and leadership, as you'll hear later, but that's for later. For now, let me leave it to thank again Christy and Josh and Noor and Janelle and many, many others who have made this meeting possible. Josh, Christy, back over to you.
Okay, wonderful. Thanks so much, Chuck, for sharing this with us. So just a couple of housekeeping items before we get started. This is a two-day event from 1 to 5 Eastern Time so that you know what to expect and when. We're going to be using Zoom to present the keynote speeches and presentations live. You'll also be able to access the session recordings on the MCBK website after the meeting. Just as a reminder, there's going to be questions and answer periods for the keynotes through the chat, so please enter any comments and questions and observations you have there during those events. And then finally, poster and demo abstracts have also been shared with you. You received an email, I believe, yesterday or the day before, as well as PDFs of the posters available for easy viewing. So please take a look at those and be ready to engage with our contributors, and a huge round of thanks to everyone who contributed to this year's program. So with that, I'm going to hand it over to Josh for our next moving into the program.
Thanks, Christy. And just for the audience, and I'd like to set the stage a little bit in terms of where we currently are and why MCBK is building the community and promoting its efforts as it is. We begin by looking at the rationale for computable knowledge and looking at the learning health system and transforming knowledge from data to knowledge, knowledge to performance, and performance back to data. So to achieve discovery through implementation. However, this is unfortunately takes, as we all know, too long a time in order to move from discovery to implementation back to discovery, and really affecting implementation cycles that improve learning healthcare system and achieve and improve learning healthcare systems. What is key to this, and this missing piece here in the next slide, is the keystone, really, of knowledge, and that is essential in the learning cycle. And the key idea is to make biomedical knowledge computable so that it is findable, accessible, interoperable, reusable, and trusted, or what we call FAIR-T. Next slide. So Chuck has pontificated about this, and others have really, I think, talked about how computable knowledge enables a second knowledge revolution. First revolution, sparked by the printing press, enabled mass access, but really a second revolution enables mass action that's necessary for learning health systems in order to move knowledge, data to knowledge, knowledge to performance, and performance back to data through these iterative and increasingly rapid learning cycles that promote increasing and more rapidly promote improved performance and improved health. And the features of mass action include the delivery of knowledge-driven interventions at unlimited scale. When knowledge changes, the implementation can be large-scale and instantaneous, and really achieve synergy among knowledge elements or objects that can generate interventions with value that exceeds the sum of their parts. And I think we're seeing this more and greater emphasis being placed upon the rapid development of large language of LLMs, of AI, and the continued work that has been ongoing in terms of implementation of all kinds of knowledge and making that knowledge, such as computable guidelines, knowledge trees, and so forth, in this second knowledge revolution. Next slide. So MCBK was formed as a multi-stakeholder movement to mobilize fair and trusted computable knowledge to improve health and healthcare. Meetings have gone back to 2017, with the initial meeting in Ann Arbor. Open in-person meetings at NIH 2018 and 2019. Moving into the UK meeting in London in 2019. And we've had open virtual meetings in 2020, 2021, and virtual global meetings in 2022, 2023. And we expect this to continue onward and upward as MCBK continues to grow. Next slide. The work at MCBK as a community has really been driven by its vision as outlined in the 2018 manifesto. And I won't read through every word here, but just the highlights. And that is key to mobilizing biomedical knowledge that can support action toward improving human health using computable formats that can be shared and integrated into health information systems and applications to efficiently and equitably serve learning and knowledge needs of all participants as well as the public good. This can ensure that the knowledge properly reflects the best and most current evidence in science to achieve through the evolution of an open computable biomedical knowledge ecosystem that's dedicated to achieving the FAIR plus T principles. Next slide. As part of that effort and growing community involvement, this meeting over today and tomorrow is the first annual MCBK North America or MCBK-NA chapter meeting. The MCBK-NA has been evolving into a membership organization of administrative base at University of Michigan, 193 charter members. It's a robust and enthusiastic steering committee, which we're very appreciative and thankful for their continued involvement and guidance. We have 3 established work groups: Technical Standards and Infrastructure Working Group, Trust and Policy Working Group, and the Health Equity and Access to Learning or HEAL Working Group. The MCBK-NA chapter organized its first meeting in July 2023, and this is our first annual meeting, and I'm pleased to report that we have 305 unique registrants to this meeting. Congratulations. As part of this meeting, I'm really excited about and part of the programming. I'm really excited and to announce the posters that have been accepted and will be presented later today. We've broadly categorized them into 2 groups: AI and machine learning posters and data and evidence posters. We think that we want to thank all of those who have submitted their really exciting work, thought, and effort that went into the generation of these posters. I'm really looking forward to seeing those presentations later today. In addition to posters, we also have demonstrations where we can see real-world applications of mobilizing computable biomedical knowledge. In order to better organize those materials, we break them out into 3 rooms. It would be great to have all be able to present in the same room together so we could all witness all the great work as a community. But thank you for your flexibility and organizing and choosing between one of these 3 exciting rooms where where these great efforts are being and will be demonstrated. As we go through the chapter meeting over the next couple of days, we think there are some questions that you might want to ask yourself, and we'd love to get feedback at points throughout the meeting. What do you want MCBK-NA to be and do? What themes and topics should be emphasized at meetings? Should meetings continue to be virtual or in-person or hybrid? What should be the work groups' existing and new priorities? And how should MCBK be governed? So think of that, put those in the back of your mind and try. We look forward to getting feedback from you at different points throughout the chapter meeting. Today's day 1 program looks like the following. We'll receive an MCBK UK chapter update So really thankful to having representation from MCBK UK. Then we'll move into our plenary, into envisioning the NLM of the future, translating data into computable knowledge. Our plenary conversation of large language models and discrete object knowledge objects. We'll take a brief break and then move into our posters and demos. And then Christy and I will wrap up the day. So with that, let's move into our update from our colleagues in the UK. I'm really pleased that we have Philip Scott here to join us and give us an update. Phyllis is Deputy Editor of BMJ Health and Care Informatics and a board member of HL7 UK. He is the Program Director and Professor of Digital Skills for Health and Care Professions at the University of Wales, Trinity St. David. Philip, I open up the floor to you.
Okay, thank you very much, Josh. So you can hear me okay? Just double checking. Great, thank you very much. Okay, so it's just myself presenting today. Jeremy and I had a meeting earlier to run through the slides, so it is a joint UK position we're sharing with you. So if I can just share my screen. Can you see that okay? Yeah, okay, good. So one thing to highlight at the outset, MCBK UK doesn't exist as a separate organisation in its own right. We operate as a group within BCS, British Computer Society, the Chartered Institute for IT. So in the UK, BCS is, if you like, the professional body for IT across all sectors, not just health. But within that, we have a health and care group. And then within that, we've constituted the MCBK steering group, which I'll explain a bit more about as we go through.
Philip, my apologies. Hello. Oh, there we go. There's the screen. Sorry. Thank you.
Okay, good. So I'm just going to briefly run through the origins of MCBK UK, which we have presented at previous global conferences, but some people here may not have seen that, so that's quite brief. Quick run through of the projects that we've worked on so far, some of our recent events in the last 12 months, what we're currently working on, and plans for the future. And I very much look forward to your questions and comments at the end. So in terms of origins, it goes back to 2019. Chuck approached me and Jeremy Wyatt, and we had a sort of collaboration anyway, and he knew of our interest in particularly clinical decision support, and proposed that we should have a conference in London, which we did. And we were able to get together all the key stakeholders across the National Health Service in the 4 different nations of the UK, as well as policymakers from NHS England and NICE, which I'm sure you will have heard of as our national body for developing clinical guidelines, as well as industry and academic representation. So we were able to publish some of the presentations as papers in BMJ Health and Care Informatics, and we have a whole series of recorded talks from that event which are available on the link there to YouTube. And by the way, in the picture on the right, that's not a younger Chuck at the top there. That's a guy who's a pathologist at Great Ormond Children's Hospital in London. So you've probably heard of Great Ormond Street. And the lady in the middle was the technical advisor to the Secretary of State for Health at that time. So that got us going, and then from that we formed a steering group, and that at that time was a collaboration between the British Computer Society and another body called the Faculty of Clinical Informatics. And I'll come back to that in terms of where we are now in a moment. So here's some of the projects we've done since then. So in 2020, we were invited by Health Data Research UK, which is a publicly funded data research group. They were running a thing called Learning Loops, which is a kind of mini learning health system. And the idea was to have, research projects which produce rapid turnaround into real world implementation, and then studying the effects of that implementation. So exactly a learning health system. And so what we did with them was advise on ways in which research projects, instead of just producing apps or instead of just publishing papers, they should be publishing knowledge objects. And guess what? None of them were. There's a big surprise. So what we did was run what we called a collaborathon. So like a hackathon, but the word collaborathon is designed to be more inclusive. That doesn't sound so off-putting. And we were using the HR7 clinical quality language as a means to build prototype representations for particular clinical scenarios, which we set out for them. And there's links to a couple of blogs there, which I wrote about the activities we did with them. Following year, 2021 into 22, we gave some advice to the NHS Scotland Clinical Decision Support programme called the Right Decision Service. That resulted in a number of artefacts which they were using internally in terms of artifacts in the sense of policy, not in the sense of knowledge objects per se. Then in 2022, we consolidated our work to date in the white paper shown on the right there, which is available at the link shown. So that was a BCS white paper setting out basically the MCBK agenda as we understood it for the UK. And then we were also invited to give some support to the NHS England Clinical Decision Support Programme, which unfortunately has stalled at the moment due to financial pressures, but we're hoping if our economy ever gets resurrected, then we might get some funding to be able to support that programme. And the biggest thing we wanted to report from projects is the work we've been doing with NICE around computable clinical guidelines. So that started in 2022 and is still ongoing. So we called the project NICE Computable Implementation Guidance, NCIG for short. The diagram in the middle gives you the main timeline up to kind of early 2023, and that's the timeline we used in the Learning Health Systems paper that's shown at the bottom right there. And importantly, we wanted to stress our adoption of the World Health Organization Digital Accessibility Kit, the DAC, which is illustrated in those two colourful diagrams at the bottom, because we found that to be a really helpful technology-neutral way of modelling clinical guidelines. So it's not an executable thing, but it's an important step towards executable representation of knowledge. So more recently, during 2023, we ran a workshop in February around regulation of computable knowledge with the appropriate bodies in the UK. So MHRA is the Medicines and Healthcare Regulatory Agency. So they primarily deal with drugs and medical devices, but they also have a role with decision support. So they're, I guess, equivalent to the FDA in the US. NICE, hopefully you've heard of, and MAAS actually doesn't exist anymore, but it was the multi-agency advisory service which was set up to advise industry, particularly industry creating AI-based products, to help them navigate the rather confusing regulatory landscape that we have in the UK. And again, we published a paper in Learning Health Systems, which is linked there. We held a national conference in London again in May. We presented about our work with NICE in September at the Guidelines International Network, which held its international conference in Glasgow in Scotland this year. And then we followed that up with a presentation in November. We held a workshop specifically about the use of document templates and e-forms as used in EHRs and specifically in primary care EHRs. As we regard that as kind of low-hanging fruit, as the kind of thing which is computable knowledge at a fairly simple level, but is also quite widely used, widely understood, and seems to us to be a good target for bringing it into the MCBK worldview. We've been continuing our fortnightly project meetings with NICE and a related body called PRSB, linked there, which is the Professional Record Standards body. So they don't produce technical standards like FHIR, they produce standards from a clinical perspective in terms of information structure and content for particular documents, communications, and records. And more recently, we've started a collaboration with the Turing Institute, which again is a publicly funded AI research group, and they specifically have a clinical AI group. as well as the AI special interest group that exists within the British Computer Society. So we're kind of triangulating our activities with them. So as mentioned, our current work, at the heart of it has been this activity with NICE. So the program that we ran with them since 2022 has really helped them change their view of what guideline development needs to look like. As you know, at the moment, all various bodies that produce clinical guidelines, they are then reverse engineered into some kind of software, some kind of logic, some kind of algorithm. And every company, every developer does it separately, does it differently. They all risk repeating different errors, mistranslating, interpolating. It's very wasteful, it's very inefficient, and it isn't the vision of MCBK. So NICE now sees that they need to be building in computability prospectively from the very beginning of guideline development. And so they have adopted at board level that they're going to change their process. It's going to start from April. And so during the first quarter of this calendar year, they're now in the process of working out how to implement that. So their aims are to build computability into all guideline production. Now, what that means remains to be defined. Does it just go as far as a technology neutral model like the DAC, or does it go further into something like CPG on Fire? They recognise the need to modify their governance structures. They need education, both for their staff and for the expert committees who work on particular specialty guidelines. And they're formalising their relationships with PRSB, as I mentioned, and other standards bodies. So that's really exciting, very much work in progress, and we hope to see those changes in effect from the new financial year. So plans for the future briefly, because unfortunately the Faculty of Clinical Informatics that we were partnering with, they no longer exist. So now this is going to become purely a BCS special interest group. We've been undertaking a SWOT analysis, so strengths, weaknesses, opportunities, threats, to understand our kind of organisational strategy, mapping out who our stakeholders are. And then from that, we're developing a marketing and communications plan. So what are our key messages? Who do we want to influence to do what? And frankly, making MCBK meaningful, because outside the geek community and even inside the geek community, sometimes people just don't know what MCBK means. We think we need a kind of a strap line to go with it to make it more readily comprehensible. And then continue building out a membership community, both within BCS and beyond. And then later this year, we're going to be contributing to the launch conference for this new structure we're calling the BCS Faculty of Digital Health and Care, which hopefully will be happening in October. And we'll be continuing to work with NICE to help and support and advise them as they move through this change of process towards prospective development of computability. So busy times ahead. So thank you very much for the invitation to come and talk to you today. I hope you found that useful, interesting. Very much welcome your questions and comments.
Thank you, Philip, for the fantastic update, and congratulations on all the great work that you and your chapter have been working on. I see we have a question from Alan. Alan.
Yes, thank you. Thank you, Phil, for that presentation. I have two questions. What are your intentions regarding development of tools that achieve personalized medicine instructions for individual patient encounters? And the second is, what are your thoughts about acquiring information that could answer the question, so what? Is this a good idea or is this a bad idea? In other words, how can we acquire scientifically credible and compelling evidence that this either works and should be globally distributed, or we should redistribute our efforts and play more football and not so much worry about computers?
Sure, no, very good questions. Thank you, Alan. On the first one, what plans we have for tools for precision medicine. None at all. We haven't scratched the surface or even thought about that, to be perfectly honest. Because we're not driven by NICE, but we are constrained by NICE, and we have zero funding. So what we're doing is trying to help NICE think through how they change their processes. At the moment, the thinking is still, frankly, on single condition guidelines. Even multi-morbidity guidelines remains a challenge, let alone personalisation. Having said that, there is, I think, some encouraging work that's come out recently about ability to detect multi-morbidity conflicts between guidelines. So, but that's not something we've done. That's something we would be looking to learn from what others have done and see if we can apply that within the NICE scenario. So that's number one, i.e. not very much. The second one about does this work and is it worth doing? Again, I didn't mention it, and it's a very good question. Within NICE's framework, they are very much looking at outcomes. So part of what they want to do is look at compliance with guidelines and impact of compliance with guidelines. So there's can we look at EHR data at a population level and detect compliance with guidelines? And then can we see any association between compliance with guidelines and outcomes at an individual patient or population level? So how that's going to happen is still to be worked out, but it's very much a live question. I don't know if that addresses the point you were quite making, but that's the area that we're addressing at the moment.
Thank you. That's a start.
Yep. And we don't claim to have more than a start. It's baby steps here.
Philip, thank you so much for the fantastic overview. If anyone has additional questions, please feel free to enter them into the chat and we'll forward them to Philip. Looking at the time, I think we're ready to move on to the next portion of our agenda. And it's over to you, Christy, to introduce our featured keynote.
Okay, wonderful. Well, thank you very much. It is a distinct pleasure and honor to introduce Dr. Richard Schuerman. Dr. Schuerman brings vast expertise in informatics, data science, and computational methods that are coupled with experience in immunology and infectious diseases to the National Library of Medicine. As scientific director, he is responsible for setting scientific priorities and for developing a comprehensive vision for NLM's broad intramural research program, which includes components housed at both Lister Hill National Center for Biomedical Communications and the National Center for Biotechnology Information. Dr. Schuerman guides NLM's ongoing efforts to align NLM's intramural research priorities with the research priorities of NIH itself and the broader biomedical research community. During his career, Dr. Schuerman has applied his deep knowledge in molecular immunology and infectious disease towards the development of novel computational data mining methods and knowledge representation approaches. These computational methods have been made available through several public database and analysis resources, including the Bacterial and Viral Bioinformatics Resource Center and the Immunology Database and Analysis Portal. So both are supported by NIH. More recently, Dr. Schuerman has focused on the development of novel artificial intelligence approaches for interpreting single-cell genomics data of the human immune and nervous systems. Prior to joining NLM, Dr. Schuerman was at the La Jolla campus director and director of the informatics at the J. Craig Venter Institute, an adjunct professor of pathology at the University of California, San Diego, and in the Center for Infectious Disease and Vaccine Research at the La Jolla Institute for Immunology. So with that, I'd like to welcome Dr. Schuerman to give his remarks. Thank you very much for joining us.
Thank you, Christy. Okay. Yeah. Thank you, Christy. And let me just start by thanking Christy and Josh for the opportunity to speak with you today. I wanted to take this opportunity to talk about a new project that we're working on at the National Library of Medicine that's, I think, very much aligned with the goals of this organization. And that is, how do we take data and converted into computable knowledge to kind of foster applications in the healthcare sector. So let me share my screen. Right. I think we should be good to go. So just to give you a little bit of a background about the National Library of Medicine, it's one of the 27 institutes and centers that compose the U.S. National Institutes of Health. And the National Library of Medicine has had a very interesting history. It started out as a collection of books, and it was initially part of the office of the U.S. Army Attorney, Surgeon General. And that's how it really started in 1836. But it's had a very interesting evolution over its lifespan, so that now it has kind of expanded to not just managing collections of biomedical journals and articles and books, but also to become a trusted source of biomedical data and other forms of knowledge that the community can use. And what has happened really in the 21st century is that this mission of managing biomedical data and knowledge has really expanded pretty dramatically. So there's many different resources that the National Library of Medicine has developed, and you're probably familiar with many of these things. From the research literature perspective, we manage PubMed and PubMed Central and Medline as indexes of the biomedical literature. We have a whole variety of different database resources and analysis tools like GenBank and BLAST, and the new comparative genomics resource at NIH. We've been involved in developing a variety of healthcare standards, and we continue to maintain those like UMLS and MESH and LOINC. And a variety of other kinds of library resources and consumer health resources as resources for the biomedical community in general. And I think the common theme behind what we're trying to accomplish is to provide trusted sources of this kind of information so that when you come to the National Library of Medicine, you can trust the information that you're obtaining. And there's a very interesting statistic that I put in this slide here about the amount of data that we're actually processing at the National Library of Medicine is on the order of 500 terabytes of data a day, kind of goes through our analytical pipelines, our submission interfaces, and things like that. And just to put that in perspective, the Hubble Space Telescope only manages about 1 terabyte of data a year. So it's just phenomenal how much data is flowing through the National Library of Medicine through these different resources. So just as an example of how we're managing some of the data that's coming in, this is an example of a record from the Gene Expression Omnibus, GEO, that is a resource for gene expression data. And what you'll see on the right-hand side here is just an example of one of the records for this data set in GEO, where we have summary information about the experiment that was performed that generated the gene expression data. And then at the bottom, you have the ability to access that data in kind of a matrix format, which basically has samples by genes and the expression level of those genes in those samples. So I think, as an institution, NLM has done a pretty good job of establishing data repositories for this kind of data. But what I think we haven't done as good a job at is coming up with strategies to capture the knowledge that is derived from the analysis and interpretation of those data. We do have knowledge information, but most of that is actually found in free text form in the publications that we manage through PubMed Central and through all the journals and those resources. And so we do have knowledge, but it's in the form of this free text. So I think one of the challenges moving forward and where the National Library of Medicine is heading is, can we come up with strategies for computing or translating big data into computable knowledge? How can we facilitate that kind of translation, and how can we do that at the scale of data that we're dealing with? It's definitely a big challenge. So what I wanted to talk to you today about is a new pilot that we're starting that's focused on trying to build a new knowledge source about cell phenotypes and their role in disease. So cells are probably the most fundamental functional units of life. As you know, different cell types play different physiological roles in the body, and that the distinct phenotype, the identity and function of a cell, is dictated by that subset of genes and proteins that are expressed. To a first approximation, all of our cells have the same genome, but it's the expressed genome that gives rise to the distinct functions of each of these different types of cells. And so that if there's any abnormalities in the expressed genome, that kind of forms the physical basis of disease. And so if we can understand normal and abnormal cell phenotypes, it will allow us to identify new diagnostic biomarkers of disease and hopefully also identifying new therapeutic target candidates. So traditionally, we have tried to understand cell phenotypes by doing transcriptional profiling of bulk samples from tissues, but that tends to obscure the complexity of the tissue, such that you tend to get information about the most abundant cell types, but not as much information about the more rare cell types that would be present in that specimen. But over the last decade or so, this new technology of doing single cell or single nucleus RNA sequencing is now allowing us to quantify and characterize these cell phenotypes in an unbiased way, so that we're able to identify both known cell subsets and novel cell subsets, even rare cell subsets in these complex tissues. And so in parallel, we're seeing now the explosion of the application of machine learning and artificial intelligence that are emerging as really extremely valuable tools to help us characterize this complexity. So we've been involved in a consortium called the Human Cell Atlas that is trying to develop comprehensive reference maps of all human cells as the basis for understanding fundamental biological processes, but also for developing diagnostics and therapeutics. It's a large international consortium, and there are a number of different working groups that have been assembled to work on specific organ systems to build these atlases. And so we have been, one of the workhorses, I think, for this approach has been the single cell or single nucleus transcriptional profiling. And essentially what this workflow looks like is that you can take a tissue specimen, you can maybe microdissect particular regions within that specimen, then isolate single cells or single nuclei, and then take that material and go through RNA sequencing using standard platforms to produce kind of a primary output from these workflows, which is what we call a cell by gene expression matrix, where in this heat map, you see that each column here corresponds to a cell or a nucleus, and the rows correspond to different genes and their expression levels in the heat map. And then typically what people do is use unsupervised clustering to group together cells that have similar transcriptional profiles, with the idea that if cells share a transcriptional profile, they're probably cells of the same type. So this technology now is exploding, and it seems like every group now is applying this kind of single cell transcriptional profiling to understand different tissues and different diseases, different perturbations, things like that. So I just did a very quick PubMed search for publications that use single cell RNA sequencing in humans. Just within the last year, there's over 2,000 publications using this technology on human samples. And so it's become an incredibly powerful technology, and there's a lot of really interesting data that's coming out of these studies. And there are database repositories that have been set up. This is one that was established by the Chan-Zuckerberg initiative called Cell by Gene, that again is capturing a lot of the data that's coming out of these experiments. We're particularly interested in the human brain, and especially the human neocortex, because this is the part of the brain that's involved in a lot of the higher level functions in mammals, including spatial reasoning, conscious thought, and language in humans. And so we're very interested in trying to understand how do different cell types kind of contribute to these higher level functions. And so we have, historically, we've known that there are different types of neurons in the brain based on different morphological characteristics that have been defined based on histology. And these are some examples of different types of cell morphologies that you see for neurons in the human brain. But once we started to look at the expression of specific genes, we realized that the complexity is probably much greater than what we suspected just looking at the morphology of the neurons. And so here, these are just a couple of examples of genes. SLC17A7 is a gene expressed in excitatory neurons. And you can see here, there's this characteristic laminar pattern of genes, or sorry, of cells within the neocortex. The excitatory neurons seem to be absent from layer 1, which is the top layer, the apical layer of the cortex, whereas this marker of inhibitory genes is found in layer 1, but it's largely absent from the deeper cortical layers. And so as we started to look at the expression pattern of these different genes, it became clear that the complexity of cell phenotypes is going to be much greater than we suspected from morphology. So we've been collaborating with the Allen Institute for Brain Science to try to bring together information from these transcriptional profiling technologies with information about the morphological structure of neurons, electrophysiological properties, and also the synaptomics, the connectivity between the different types of neurons, to develop kind of this data-driven gold standard taxonomy of the human neocortex. So again, this is kind of the standard profile that we use. And in the first big study that we did, we looked at one brain region called the middle temporal gyrus. And when we took the single cell transcriptomic data and did unsupervised clustering, we defined about 75 distinct types of cell phenotypes. Most of them are different types of neurons. And on the right-hand side here is a heat map that shows the expression of historical markers. These are known genes that were known to be expressed in human brain, in specific types of neurons in the human brain. And the columns here correspond to the different cell types that were defined by this unsupervised clustering. And the dendrogram reflects kind of the relatedness between the different cell types. And so at the highest level here, you can see there's a subdivision between inhibitory neurons and excitatory neurons. And then over here on the right-hand side are the non-neuronal cells, the astrocytes, oligodendrocytes, microglia, et cetera. But now if you look at the individual genes here, this really reflects prior knowledge about cell types and the genes that they express. And what you could see, for example, in the middle part here of the inhibitory neurons, we know that they express GAD1, and this particular type of inhibitory neuron expresses this VIP gene here. But we see that there's about a dozen subtypes of VIP-expressing inhibitory neurons that we really had no understanding of that complexity. And we didn't have any knowledge about what genes might distinguish between these subtypes of VIP-expressing inhibitory neurons. So my group decided to develop a computational method to identify biomarkers for these cell types using random forest machine learning that we call NSForest. I don't have time to really kind of get into the details about how the algorithm works, other than to say that we realized that some of these machine learning methods that are kind of retain some level of explainability in how they build their predictive models. Those features that are used for models can be useful for identifying very specific markers for these different cell types that we are identifying here. And so here's just now an example of we take all of this transcriptomic data and we pass that through this random forest algorithm, NSForest. And now here's the expression of the marker genes that comes out of that. And you could, what you would like to see from this kind of analysis is this nice diagonal pattern of expression where that the marker genes are expressed exclusively in cells of one of these types and basically not expressed off diagonally in cells of other types. But I think there's a lot of really interesting biology that's going to come out of this kind of analysis. And so on the right-hand side, I have one example of this. So it turns out that a lot of the very specific markers for these different subtypes of neurons are actually long non-coding RNAs. And at this stage, and that's what I have highlighted with these red arrows on the right. So we don't really know what role non-coding RNAs are playing in cell type identity here, but they are very specific for certain types of neurons. It's likely that they're playing maybe a regulatory role and regulating, the expression profiles of these certain types of neurons, and maybe they're kind of playing a complementary role to transcription factors that might be specific for different subtypes of neurons. But I think this is just one example of kind of emerging knowledge that's going to come out of this kind of work. So we're now in the process of trying to apply this to the whole mouse brain and the whole human brain, which is a huge big data challenge, I can tell you. This is a massive amount of data, and the computational complexity of some of these methods is pretty challenging. And it looks like what we're dealing with is probably on the order of 4,000 to 5,000 different types of neurons in mammalian brain. And so, our challenge is to try to come up with ways of very specifically, recognizing these different types of neurons using specific markers. So what I want to do now is get to this knowledge representation part. So what can we do with the marker genes? Well, there's a lot of different things that we can do with them. But one of the things that we want to do is use them as a way of developing semantic definitions and knowledge representations of these cell phenotypes. And so we've been involved in building an ontology called the cell ontology, which is the objective is to develop these semantic representations of cell phenotypes and make that available for the community. And so what we proposed is that can we use this kind of information from these single cell transcriptomic experiments to develop a semantic definition of cell types? And the way that we would do that is we would combine information about where the samples are coming from, the specimen sources, that would include the anatomical structure and the species that the specimen was derived from. Is there a parent cell class that's already in the cell ontology that is kind of a higher level phenotype for groups of cells that have similar characteristics? But then as we get to this very specific level, can we then use these minimum set of necessary and sufficient marker genes as a way of very specifically defining these cell types? And so with that structure, we can then come up with a definition for a cell type that might look something like this. A human middle temporal gyrus cortical layer 1. So that's kind of the source of the specimen. It's a GABAergic interneuron. That's the parent cell class that's already in the cell ontology. But this particular type of GABAergic interneuron selectively expresses these marker genes. And so now we have a very precise semantic definition of this cell type that we can then use to put into the cell ontology. And so this is an example of what this kind of information looks like. So here is one example, and basically it's kind of very similar to what I just described. But now what we can do is we can start adding into this cell ontology all of these distinct cell types that are being identified through these single cell transcriptomic experiments. So, okay, so the other thing that we can then do that is produce different representations of that semantics. So now here is again our marker gene expression, and we can then use this to construct semantic triples of subject, predicate, object, which are amenable to loading into representational approaches like graph databases. And so basically what we've done here is we've used this Cypher query language that is supported by the Neo4j graph database infrastructure. And we can take the marker gene expression profile and convert it into this graphical representation of that as a form of knowledge about those cell phenotypes. And so, we have methods to be able to do that. But I think the real power of doing that kind of an approach is that we can then start thinking about, well, let's integrate this information about cell phenotypes with other information that we have about genes and genetic predispositions to diseases and drug target information with the proteins that are encoded by those genes. And so we can start to think about, here are a bunch of the nodes that would the different types of data that would serve as nodes in this semantic graph, things like cell types, organisms, anatomical structures, proteins, protein functions, cellular locations, drug compounds, diseases, signs and symptoms of diseases, mutant alleles. And then we can put together these semantic triples of subject, predicate, object, where we can connect cell types with anatomical structures. So this cell type is part of this anatomical structure. This biomarker combination is a marker for this cell type. This protein is part of this cell type. This gene produces this transcript. This mutant allele is a genetic basis for a condition, a disease. And this drug compound molecularly interacts with this protein. And so now you can start to see, by having these standard node representations and standard relationships, we can start to integrate all of this knowledge that we've been accumulating over the years about genes and proteins and diseases and cells and things like that. So this is now what this kind of graphical framework would look like, where we're bringing all this stuff together. And so now there are several groups that are assembling these integrated knowledge graph representations by bringing together resources like many of the resources that we manage at the National Library of Medicine, like OMIM and PubChem and Gene and GenBank and places like that. And so now what we can do is, represent all of those things in this kind of semantic graph. And then can we integrate that with all this new data that's coming out of the this new technology, the single cell transcriptional profiling. So we did that, and this is just an example of what can emerge from that kind of integrative approach. So what we did here is that we pulled out a subgraph of all of these kinds of nodes of entities that are connected to Alzheimer's disease. So here's Alzheimer's disease in this red node, and here are all the things that are connected to Alzheimer's disease through these structured relationships. And what we found is that there are certain cell types that have a very interesting connection to Alzheimer's disease. So there's this particular type of inhibitory neuron here that expresses, there's one marker gene that specifically tags that type of inhibitory neuron. That marker gene has been genetically associated with Alzheimer's disease in a GWAS study. And it also turns out that the protein that's produced by that gene is a target from an Alzheimer's therapeutic compound called galantamine. So now we see that this particular cell type is connected to Alzheimer's disease in two interesting ways. The marker gene for that cell type is associated with genetic predisposition to Alzheimer's, and the protein produced by that gene is also a therapeutic target. So I think this illustrates an example of what can emerge if we can come up with a way of taking these large data sets that are coming out of these kind of high throughput experimental techniques and coming up with a standard translational approach to convert that into a semantic knowledge representation that then can be integrated with all this other knowledge that we're managing. And so we've started a new pilot to do this at NCBI that we're calling the NCBI Cell Pilot, to take all of these data sets that are available in these public repositories, process them through standard pipelines to produce these semantically structured triple assertions to load into a graphical database representation, and then build user interfaces that allow the community to then interact with that data, extract information, extract the knowledge that's embedded in that knowledge graph. But at the beginning, I talked about all this knowledge that's in the literature. We don't want to ignore that either, right? So the other half of this project is, can we now take advantage of these new artificial intelligence approaches, the large language models that could be applied for natural language processing, to extract other information about cell phenotypes from the free text of these articles that are being published about the interpretation of these data? Because in the results section of these papers, you have the subject matter experts have looked at the data and they've interpreted it, and they've extracted what they think is the most significant information from that data. But that's right now buried in this free text. So what we're doing in parallel then is to try to apply these natural language processing methods to extract the same kinds of information in the same kind of structured semantics from the free text, so that we can load it and integrate it into the same knowledge graph representations, so that we can now get the best of both worlds. We take the data-driven approach for knowledge extraction, and then, the knowledge extraction approach using natural language processing from the text. And then this will now also allow us to access all of that prior knowledge in the literature that we manage at the National Library of Medicine. So that's the goal for this pilot. These two streams of knowledge, one the data-driven stream, the other kind of a knowledge extraction stream from the literature. And hopefully we'll be able to build a cell type knowledge base that can serve as a paradigm for establishing other kinds of knowledge bases for other kinds of information from other kinds of experimental technologies about diseases and proteins and metabolites and things like that. So we're hoping that this is a paradigm for how we can extract computable knowledge from big data. And so this is an example of what the interface might look like, et cetera, but you can kind of imagine what the goal would be. So let me just stop by acknowledging this has been a terrific collaboration with Ed Lean's group at the Allen Institute for Brain Science, and also the European Bioinformatics Institute, especially David Osumi Sutherland and his colleagues. And now there's a growing team at the National Library of Medicine that we're assembling to try to tackle this challenge and produce this resource. So thanks for your attention, and I'd be happy to take any questions.
Okay, can I turn my?
Yeah, let me stop sharing.
I'll
turn my mic off. Okay.
Okay, wonderful. Thank you so much, Dr. Schurman. I really appreciate it. And I think we all appreciated getting a glimpse at what's happening at the National Library of Medicine. I think it goes without saying, but I'm delighted to reinforce the incredible science that's happening both through extramurally supported research as well as through the intramural program. So getting a chance to see that is really such a treat today. So I do notice that there's a question in the chat, and then I have a couple of questions as well. So our first question, and actually Richard and I are in the same room, so we're switching between microphones and being on and off. So thanks for your patience as I'm doing that transition myself. Our first question is from Alan John Flynn, who's wondering how dynamic cell phenotypes are. So how dynamic are cell phenotypes? Simply put, can a cell change its type? And so I'll hand it over to Richard.
Yeah, that's a great question. So we've been having this philosophical debate about the distinction between cell types and cell states. And I think there are some ideas. So we have an intuition that there's a difference there, but coming up with a clearer distinction about what's a cell type and what's a cell state, I think is still a little bit of a works in progress about how we can make that distinction. My personal feeling is that cell types, once a cell attains a particular type, that's largely irreversible under normal conditions. Whereas cell states, I think, can be acquired in a reversible fashion. So like, for example, a T cell can become activated, and then when that stimulus disappears and its environment changes, it then can resume a resting state. I think this notion about the transition of cell phenotypes is a really important one, both from a developmental perspective as well as how cells respond to changing environments. And I think the knowledge base is going to have to be compatible and be able to represent that kind of dynamic feature of cell phenotypes under those kinds of circumstances. So it's a great question.
Okay, I do have a question. So I'm incredibly interested about this concept of integrated semantic knowledge graphs. And I'm wondering, so we're getting to think, it's kind of funny because I was writing questions that were coming up and Dr. Schurman was answering them as I was thinking about it. But I'm left with one question about this work, which is, so what's the next step? We think about how, in the context of this cell atlas and the cell data, we can begin to think about how it's related to all of these other concepts. And we saw that in Dr. Schurman's slide about edges and nodes and thinking about that interrelatedness. And I'm wondering, as you are integrating all of these disparate concepts and connecting them, how do you integrate those knowledge resources and also make the connections from past to future? So maintaining that line of evidence or translation of knowledge into discovery moving forward.
Yes, I think there are two aspects of that question. One is, how do we leverage these existing resources and convert that information into these semantic representations? And then the other is evidence, which is a big issue. So, if you use the NCBI resources that the NLM manages, there are a lot of points of integration of these distinct resources, but they tend to be link outs, right? It's all focused on linking some reference to an entity in this database with some reference in another database. But it's just a single link. It's not a semantic relationship. So we're starting to have discussions about, how can we add that layer of semantics on those linking structures that were within the resource. And if we can do that in a way that is kind of semi-automated so that we can actually apply it at scale to these resources, then I think we can start to build semantic representations from these existing resources, which would be incredible. The question about, managing strengths of evidence or sources of evidence when we're making these assertions, I think that's another really important aspect of the approach that we want to take, is that we want to be able to assign evidence to the edges because we're making these assertions about these specific kinds of relationships. Where does that evidence come from? Is it quantitative? Is it statistical? Is it, like expert knowledge, those kinds of things. And then it would be nice to be able to filter a subgraph based on the strength of evidence, right? You may be very liberal in what you want to accept, or you might be very stringent. So I think that has to be part of this approach, is we have to capture evidence at some level, hopefully in a computable way.
Okay, wonderful. I do see that Alan Morris has a question. Alan, would you like to ask your question?
Yes, thank you. Thank you, Dr. Sherman. That presentation makes science fiction pale by
We're
comparison.
not hearing you, Alan.
Can you hear me now? Can you hear me now?
No.
Oh,
I can hear
somebody can
Alan
hear.
if it's
I hear
helpful.
him fine.
oh, so it's not me.
There it is. Okay, sorry.
Oh,
Now,
okay.
now we can hear you.
Okay, so there's a delay time. Anyway.
No, it's
Thank
user
you.
error. I had muted you. I'm sorry.
You've just made me feel very, very much better. Thank you so much. That presentation makes science fiction pale by comparison. Thank you so much. My focus is on how knowledge will aid decision-making at a different scale, at the clinician-patient encounter scale. So I have two questions for you. One is, what are your thoughts or plans to deal with the heterogeneity of expression of disease states, like metastatic cancers that have different expressions in different metastases in different parts of the body? That must be a general biological issue that will be represented in other conditions as well.
Mhm.
And then the second is linked to previous questions, and that is noise. The clinical databases, the electronic health records, are extremely noisy and incorporate much systematic noise, which is particularly problematic. I understand that that's obvious to many people on the call. How do you plan to deal with that, as well as the noise in the literature, even without considering predatory journals? I mean, so much of the literature is of a quality that doesn't justify incorporation. How will you address those two issues, the heterogeneity of expression and the noise in the information?
Yeah. So, this notion of variable expressivity is something that is, it's a concept that we have to come to grips with in the medical community in general. And, we're starting to see that in terms of diseases that, maybe something that we've always considered to be a single disease is actually maybe 4 different subtypes of endotypes of disease. And that part of the variable expressivity may be related to endotypes. But I think disease progression, even of a single disease, can be variable, and it can be impacted by genetic factors, environmental factors, et cetera. So I do think that, the knowledge framework also has to be able to deal with those issues of variable expressivity, whether it's at the gene expression level or at the symptom level or whatever. And we have to recognize that that is a part of disease progression. So we have to deal with that. In terms of the noise in the data and the noise in the literature, that's something that we've had to deal with even before the application of these computational methods, right? We all use our own expert knowledge to try to distinguish, the important wheat from the chaff. So I don't think it's a unique problem to the computational methods, but I think we have a big advantage and an opportunity, because by applying these kind of large-scale computational approaches, we can do kind of a large-scale analysis of all the information that's out there And, we can then say, well, if something only been reported once or twice, we should take it with a grain of salt. But if many people have seen the same phenomenon, in complementary approaches, complementary studies, then I think I would become more confident that this is a real phenomenon that's being described. So, I think those are some of the thoughts about how we can, leverage these computational approaches to deal with things like noise.
Thank you.
Turn mine on. Okay. Wonderful. We are at the end of our time together, but I do see another hand up. So if I could please ask you to put your question in the chat, and then we'll make sure that it gets answered. And I'm also going to add my other question for Dr. Sherman to the chat as well, because this is really just such a fascinating topic. Again, Dr. Sherman, thank you so much for joining us today. This is such a bright and exciting vision about really taking that information or data and turning it into actionable knowledge and making that possible in ways that really haven't been possible before. So thank you so much for joining us. And with that,
Thanks, everyone.
To turn it over to Josh.
Thanks, Christy, and thanks again, Dr. Sherman, for your enlightening presentation and great discussion. And thanks to all who've entered questions and comments in the chat. Keep them coming. They're really fantastic. We'll now segue to a plenary conversation led by Dr. Mark Musin on large language models and discrete knowledge objects. I'm reminded by Noor that this is a two-part presentation. First part being a pre-recorded conversation that Dr. Musin had with Dr. Karen Verspoor around the LLMs and discrete knowledge objects. And then Dr. Musin will expand upon that initial recording to present additional thoughts and takeaways, and really lay out for you and extend this conversation around LLMs and discrete knowledge objects. With that, let me introduce Dr. Musin, who is a professor of biomedical informatics at Stanford University, where he is director of the Stanford Center for Biomedical Informatics Research. Dr. Musin conducts research related to open science, data stewardship, intelligent systems, and biomedical decision support. His group developed Protégé, the world's most widely used technology for building and managing terminologies and ontologies. He is principal investigator of the National Center for Biomedical Ontology, one of the original national centers for biomedical computing created by the U.S. National Institutes of Health. Dr. Musin is principal investigator of the Center for Expanded Data Annotation and Retrieval, or CEDAR. CEDAR develops new technology to ease the authoring and management of biomedical experimental data and metadata to make online data sets more findable, accessible, interoperable, and reusable. Dr. Musin chaired the Health Informatics and Modeling Topic Advisory Group for the World Health Organization's revision of the International Classification of Diseases, ICD-11, and he currently directs the WHO Collaborating Center for Classification, Terminology, and Standards at Stanford University. Dr. Musin has been elected to the American College of Medical Informatics, the Association of American Physicians, the International Academy of Health Sciences Informatics, and the National Academy of Medicine. He is also founding co-editor-in-chief of the Journal of Applied Ontology. With that, Noor, if you could start up the initial piece to this plenary conversation.
You've asked Karen and me to get together and talk about the whole issue of large language models and where they stand with respect to the MCBK initiative. I guess when you look around and you see almost every human activity right now, there's concern that natural language processing somehow is going to put an end to life as we know it on the planet in terms of all human endeavors. I don't think that's the case. It's certainly not the case with MCBK. And Karen, as an expert in natural language processing, certainly has strong opinions on this topic, as I do. Karen, is the world safe?
Yeah, the world is definitely safe. No intelligence actually lurking in that machine.
I think it's helpful actually when we think about what the role of natural language processing is, and LLMs in particular might be. It's helpful just to think about what MCBK is all about. For a lot of people, it's just a bunch of letters rather that sort of gloss off the tongue. We think about whether something is mobile, whether it's computable, whether it's biomedical, whether it's knowledge. And from my perspective, I think we can view LLMs in each of those dimensions and sort of decide whether they actually fit the MCBK parameters or not. What is your sense, Karen, about mobility?
Yeah, mobility is an interesting one, right? Because if we're talking about large language models or other foundation models, these things are massive, and they, have terabytes of parameters and space in data centers that they need. So they're really not mobile in any kind of meaningful way. Now, of course, we know that we can bring data to these models, and we don't necessarily have to move the models around themselves. So that changes the notion of mobility. Certainly, they're accessible from pretty much anywhere because they're literally just a network connection away. And so, in principle, they can be leveraged in mobile ways, but clearly they're huge. I think if we look at some of the sort of the details of what this community means by mobilized, one of the points is encode once, use repeatedly. Definitely large language models fit that paradigm, right? We're encoding all of this text and this knowledge that's captured in text in a statistical or neural network model, and we can access that information over and over again in all sorts of creative ways through different kinds of prompting. On the other hand, I think one of the mobilized elements is tracking the evolution of knowledge. And that's where I think there's a much bigger question mark, because these large language models effectively are static models. They can produce dynamic outputs, but the once they're pre-trained, and it's kind of captured in that model. And unless we start then essentially putting version controls around large language models and then doing diffs between them and somehow having mechanisms for understanding how the knowledge that's captured in them has changed, we probably can't do that. So, Mark, I don't know. What is your perspective?
No, I agree with you. And I think what matters is not whether the whether the knowledge is itself mobile or whether the access is mobile. And I think we all agree that there's no difference here. And I think particularly when we think about what's happening on this side of symbolic AI, where people are creating knowledge graphs of billions of triples. Those aren't exactly mobile either, and the goal is to make them accessible from a variety of locations. So I think the mobility issue is just as cloudy in the area of symbolic decision support as it is in the case of LLMs. I think when it comes to computation, I don't even want to define computation for our purposes here.
Yeah, I mean, computation can mean lots of different things, right? And I know that we've often, certainly with symbolic AI and kind of traditional knowledge representation, we've been thinking about logic and being able to sort of do deductive closure or transitive closure, just sort of thinking through the logical implications of the knowledge representation. We can't do that kind of computing with these large language models because the knowledge is distributed in the representation, in the mathematical representation. But as you pointed out, at scale, like with these massive knowledge graphs or other kinds of formal knowledge representation, how can we really, in any kind of detailed way, follow through the logical implications of that knowledge? It's very tricky in both cases.
And just to digress for a second, it's interesting. When you think about what led to the advent of symbolic AI in the 1970s, people were really concerned about the transparency of statistical methods for doing decision support. There was a lot of angst about rule-based systems at the time and if they would be sufficiently able to escape brittleness, leading to situations where there's not enough insight into the mechanism by which they operate so that people feel confident in the results of such systems. If you look at a large knowledge graph, most people have no clue as to how such a large system is possibly able to reach conclusions. It's the same way that we interact with LLMs and have no idea why it makes the recommendations or it makes the statements that it does. And so in some sense, that whole area of brittleness has come back to us in spades 30 years later, and we don't really have a good handle on it at all.
No, although it's interesting, Mark, because I've observed that even a year ago, before OpenAI kind of blasted onto the scene and changed the whole public perception of what's happening with AI and language modeling in particular, there was a big emphasis on explainability. So, when people were talking about the use of AI in medical decision-making or clinical decision-making, they were very emphatic. No, it needs to be explainable. We cannot possibly have a decision support tool that isn't explainable, that isn't able to provide a context for a particular decision. That I've seen that conversation actually fall by the wayside, because what's happened is the large language models and the generative systems that we have in things like ChatGPT are so convincing linguistically, and the level of competency that's in them, that people believe them without needing an explanation for why it's true, because I think it's expressed in language that they can relate to and ascribe meaning to themselves. And so that kind of need for an explanation is either provided in the response itself or implicit, because kind of the words make sense in that context, and we don't need further justification beyond those words.
That's really fascinating, because again, when you think about the advent of clinical decision support, people were convinced that we needed AI, and in that sense, it means symbolic AI, because physicians would never accept any kind of decision-making on the part of computers without the ability for the computer to explain its results. Bayesian approaches, statistical approaches, just were not going to be acceptable because they didn't have that element of explanation. And now, fast forward, and here we are. We have knowledge graphs that are so big, no one can understand what they have in terms of their scope of knowledge encoding. And LLMs, we'll just excuse them for hallucinating now and then without having any ability to really understand whether they're telling us the truth or not. It's like suddenly the sine qua non of intelligent systems is gone.
Yeah, no, that's right. And unless we do kind of rigorous fact checking and propositional truth inference modeling, even on those large symbolic systems, we don't really know for sure that the inferences are valid. And you can control for that to some extent in a symbolic system. We can't control for that in a language model, but we have other strategies for essentially cross-checking the information that's provided by these large language models, which we don't have in the symbolic systems. So it's an interesting world we're in.
Yeah, I just want to recapitulate. We've just said that there's no difference in mobility with respect to current symbolic approaches and language models. We said that they're equally computable. I don't want to argue biological. I think we both would agree that we can put biological knowledge into any kind of formalism like that. Are they knowledge, though? What are LLMs representations of knowledge?
This is the $1 million question, isn't it? So, from my perspective, large language models certainly don't encode semantic relationships, right? So fundamentally what we're capturing in these models is statistical co-occurrence patterns. Now, very sophisticated ones, and obviously using an incredible amount of context so that we can capture really subtle nuances in those co-occurrences. but it is fundamentally just co-occurrence. And in fact, the task that we're training a generative system for is really just a next word prediction task. And so there's no inherent representation of relationships. There's no semantics that's captured there in any explicit way. But what is co-occurrence? Co-occurrence implies that there's some semantic coherence between the words. And the reason words make sense together in a context is because there are connections between them. And so it really starts to raise this question of what is knowledge, right? And, do we need to have kind of formal representation of entities and relationships between them in order to say that there's knowledge? I'm not sure. I personally really believe in the power of semantics. I think one thing that is missing from these large language models is abstraction, thinking at the level of concepts rather than words. You can infer semantic relationships from the information that's encoded in these large language models. So you can work out that cats and dogs are related to each other in some way. Maybe they're connected to the notion of pet, but there's no explicit representation of that. And I think that's one really important difference between what, we as people kind of naturally do and the way we think about knowledge representation and what's in these LLMs. Where that leaves us for deciding whether or not they can contain knowledge, yeah, it's there. It's just implicit.
Well, I kind of grew up with Alan Newell's definition of knowledge, saying that a computational intelligent agent has to have goals, has to have a way of achieving those goals, uses knowledge to bring itself closer to the goals that it believes are the ones that it wants to achieve. I look at an LLM, I don't know what its goals are. And can you have knowledge without goals?
That's a tough question. So really, you're 100% right. Large language models definitely don't have goals. I mean, a generative system, the only goal that it has is to predict the next word in the sequence. At least in the manifestation of ChatGPT. There's probably other ways of realizing that in other kinds of sequence to sequence models. But even in other kinds of sequence to sequence models, where we're taking a sequence as input and then producing a sequence of words as output, it's still definitely not capturing semantics or meaning. And is that a goal? No. I mean, I think it's a very impoverished goal, if it is a goal, and it's certainly not one about problem solving beyond just trying to put the right word one after the other.
Yeah, I mean, I always used to doubt as to whether answering a question is a sufficient goal to call something intelligent, though obviously LLMs do that really well.
Yeah, and I think you're right. I mean, it's certainly not working towards a higher level objective, which I guess, relates to my earlier comment about not having concepts and relationships between concepts and being able to sort of reason in that more abstract space. They're not working towards goals. They're not meaningful goals, I guess, and they're not really kind of abstracting away from the details of the individual words. I often say to people, we're having a conversation here, Mark. Everybody who's listening to us will not remember exactly what words we said. What they're going to remember is the ideas that we conveyed and the sort of higher level messages. Where's that in a large language model? I don't think it's really there at all.
On the other hand, just abstracting, I think you and I would probably both agree that we could take a large language model, encapsulate it in some metadata that would describe what that model is good for, and make it accessible through some system where it would actually behave in an MCBK kind of way. It might not be the kind of symbolic package of knowledge that we've been talking about in this community for the past few years, but it would certainly be a good representation of knowledge, and it would solve a particular purpose.
Yeah, for sure. And I'm thinking a lot lately about essentially narrow AI, right? Trying to build on the foundations of large language models or other image-based models, foundation models more generally, and then adapting them to specific applications. And what you're referring to there in terms of metadata and trying to capture what these systems are designed for is connected to this idea of adapting them to or applying them to very specific kinds of problems. And I think actually when we're thinking about the use of these models in real world applications, that metadata will become hugely important because it needs to help us define the boundaries of use of the systems. And so we're not just talking about large language models in general, we're talking about large language models that encode knowledge or represent knowledge in some form, and then we apply it for a particular kind of decision or a particular kind of context, and specifying, what the expected use cases are for a given application is really important to have trust in it and to understand that, we've evaluated it, we tested, we believe it works in this context.
But where I have trouble is I think of a lot of symbolic representations that have got a lot of use in this community, but that have validity in various ways, but may not have generalizable intelligence, and obviously do not pass the Turing test. And then I think about large language models, which seem to pass the Turing test without really trying.
Yeah, no, it's really true because there's so much knowledge that's encoded in the large language models, and because they have that amazing linguistic competency. If we take the Turing test kind of at face value, yeah, I agree. They definitely appear to pass the Turing test, and yet I know that these machines are not intelligent, right? I've been trying to remind people that these machines are really simulating intelligence, and they're doing a really great job of simulating intelligence. But are they actually intelligent? No, I don't believe so. And maybe it comes back to that point about goal-oriented behavior and really trying to solve problems. But it just raises another question, which is, okay, if you have a machine that can pass the Turing test, then do we have a better test of intelligence? I don't know. I think we're probably not the ones who are going to solve that problem.
Well, what do you think Turing would think of a large language model?
Oh God, I think honestly he would be impressed, right? Because I mean he was the one who kind of came up with this notion of trying to get machines to encode intelligence, and so I think he would be impressed. Now, whether he would join the camp of people that are in the sort of AI, existential camp of these machines becoming our robot overlords, I don't think so, because I think he would understand that it still is a machine that we control, and maybe it would cause him to rethink his test.
Okay, so if I can summarize what I think we've said, we're saying that we're really no more intelligent than we were 30 years ago. We're still no more explainable than we were 30 years ago. We're still as brittle as we were 30 years ago, yet we feel we've made a hell of a lot of progress.
Yeah, right. The machines really appear to be much more intelligent. And, to be fair, because of the size and scale of these machines, they have incorporated a vast amount of human knowledge, more than probably any one of us can fit in our, limited neural networks. And so that scale combined with the linguistic competency of the models really, really gives us a knowledge representation that's beyond all possible previous knowledge representations. And that's what I think is really grabbing people's attention. It becomes up to us now to think about, the how we can use them effectively, under what conditions, and then, going back to that metadata and where we started, maybe, we want these things to have some constraints and boundaries associated with them. And that means describing them in computable ways, which then define the ways in which we can use them in other systems or decisions.
Agree completely. This has been fun. Thanks.
Thanks, Mark. Great.
Wonderful, thank you. And let's hand it over to Mark for additional thoughts and observations from that recorded session. Mark, I see you're off mute now.
Thanks, Josh. No, it's a pleasure to be with you live after being with you on tape. The tape that you heard really emerged from conversations that took place almost a year ago, when there was so much angst in this community as to whether LLMs were suddenly going to make MCBK obsolete. And the thought was, well, if we have all these LLMs unleashed on the world, do we need computable biomedical knowledge in any form because we have LLMs to solve all our problems? I think in some sense, we've become a lot more rational about what their strengths and limitations are over the past few months, but it's still pretty impressive what they achieve. And I think the issues that Karen and I talked about are still fairly relevant. I think it might be helpful for all of us to think about where we come to sit on the fence regarding this very issue. And Chuck, I know, composed a poll that he wanted us all to take. And I'm not sure, Josh, if you're ready to administer that or you know how to do that.
I think we have that poll ready to go, and I rest on the expertise of Janelle and Noor for doing that. And here it is. I think I see it. All feel free to please provide your responses to this multiple choice poll here.
And let me editorialize a little bit. I think most of us kind of know what an LLM is. We use the term in this poll goal-based model to be a non-LLM, a goal-based model being a more traditional decision support technology that has the goal of solving some problem, as opposed to an LLM, which is more open-ended, that will answer your question, whatever your question happens to be. Not answering a question certainly is a goal, but most of us have a better idea of what we mean by modular biomedical knowledge when we think about systems that are addressing particular challenges in biomedicine. And so when we say goal-based system, we're really meaning a system that is really constructed with the idea of addressing a particular task.
Let's give our audience another few seconds here to log their answers, and then hopefully we can take a look at the responses and react to that. Let's see here. Noor just wanted to check. Will you be showing? Oh, I see in the chat here that we have 63% response rate so far. Maybe another few seconds.
Yes, I can end it whenever, but we still have responses coming in, so I'll give it a couple seconds.
Wonderful. Thanks, Janelle.
Our goal for the rest of the session is really to see what the results of the poll are, to discuss it, and also get your views on what this community thinks is the future of LLMs and how they fit into the MCBK manifest.
Maybe while we wait for those results. Oh, and here they come. So it looks like dominated by LLMs, 11%, a mix of LLMs and goal-based models with LLMs in the lead, 46%, vice the other way around, 46%. Boy, that is a tie right there. And then dominated by goal-based models, only 1%.
Okay. There's a lot about this community, and obviously a certain amount of information about who would want to participate in this session anyway, but that's very helpful. No one thinks we're going to be taken over by LLMs, which I guess is pretty good. Well, 11% if you do. What are your thoughts? I mean, when I spoke with Karen, my belief was that I did not view LLMs as being all that different in their role than traditional knowledge-based components, that you can take a question and answering machine and package it up and make it available in a mobile computable way. And that's just as valid as something that, for example, calculates acid-base balance. How does this community weigh in on that? And I guess you can all unmute yourself as necessary. And I don't think we necessarily need to call on people unless we become totally out of hand.
Well, we have a couple hands raised already, so we might start with them, Mark. I see Alan
Okay.
had his hand up first, and so Alan, do you have any response to Mark's question, or maybe you had a question on your own?
Yeah, I was, thank you. I was wondering what Mark thought when he raised his comments about explainability with the challenges 30 years ago of symbolic representation. I wondered what his thoughts were about rule-based systems or physiologic models, both of which are explainable by examination of the elements. And with regard to LLMs, I'm just concerned that an LLM that was run several hundred years ago during the Salem witch trials might have produced some pretty powerful conclusions about witches. And just want to point out that the context within which we run them is an important determinant of what's produced.
Yeah, I agree with that, Alan, and there's no question LLMs are trained, and we don't have a lot of insight into what the training necessarily is. When rule-based systems were at their peak, which was, I guess, the early 1980s, there was this belief that they were going to be supplanting statistical systems, where people could not look at calculations and understand how those calculations could support logical conclusions. I think the challenge I always had with rule-based systems is that they can explain themselves by indicating what rules got fired under what circumstances. But the rules themselves don't necessarily tell you how to interpret the system's behavior. And just because you have a rule that says that, I don't know, if a patient is a compromised host and the patient likely has Pseudomonas infection, that's a rule that certainly is inspectable and it certainly is verifiable in the literature, but we really don't know whether that rule was invoked in the context of the rule-based system in the most appropriate way, and whether there's actually evidence for, say, the patient being a compromised host in the first place. So there's a whole cascade of doubt that is always there when looking at rules in a rule base out of context that makes the explanation as sometimes as difficult to understand as trying to understand how a Bayesian net would conclude the probability of some disease. So I think many of us feel better about rules than we do about more statistical models, but I'm not sure that rules solve the problem altogether. But I agree with you completely that when we turn to LLMs, we don't know where those LLMs come from. We don't know what text they were trained on. And as you suggest, if the text that they were trained on is archaic, then we're going to get archaic responses.
Yeah, just to clarify, Mark, when I said rule-based systems, I was referring to personalized medicine returns from rule-based systems that are actually used, tested, and validated in clinical environments. So the explainability is tightly linked to the clinical outcome, and their compelling data drive decision-making.
Right. And I agree with you that having some modicum of explanation has got to be better than having none. On the other hand, we've certainly seen machine learning skyrocket as the preferred method for building intelligent systems in AI in the past 20 years, and no one seems to care. So it seems a matter of convenience as to whether explanation is really going to be a driving factor or not. I would much rather my Tesla know the rules of the road and be able to show me the driver's manual when it makes a decision about how to drive autonomously than what happens now, where it makes decisions based on machine learning algorithms that I have absolutely no insight into. But this is the world that we have.
I just want to point out here that we have a fantastic chat going on with some amazing points and retorts. Keep it coming. This is really enjoyable. Hands up, I see Richard Shoreman. Richard.
Hey, Mark.
Long time no see.
I know, yeah. I was at the AMIA conference in November, and I was really struck by the number of presentations talking about the use of different AI techniques to develop, improved clinical decision support systems. But each one was kind of optimized to solve or address a particular use case.
Yeah.
And I was concerned that we're going to have thousands and thousands of models that have all been, optimized for very specific uses. How are we going to approach the challenge of validating all these models to actually be able to use them for clinical decisions?
That's a great question, because I think what we've gotten ourselves into is a situation where building such decision systems really is a matter of applying a commodity. It's so easy now to take some clinical problem, reduce it to a set of measurable inputs and a bunch of output classifications, throw it into a neural network and see what you get. And you end up with something like, say, the EPIC system for sepsis diagnosis, which was heralded with great fanfare and ultimately led to disappointment. And I'm not sure what we have to validate that. We don't have a clearinghouse for decision support technology. About 30 years ago, 30 years? Yeah, about maybe 25. There was an organization called MCBK, which was basically an organization trying to create a catalog of all AI systems and try to become like a consumer reports for all these systems, which is a lot more reasonable when you had only a handful of such systems being promoted at any particular time. Still, it was an organization that was funded with money from the drug companies, and MCBK, as it was called, ultimately died because people didn't trust it. Now, if we were to do that, we'd have the problem of having an order of magnitude more systems that people might want to evaluate. And then the question becomes, geez, how do we know how to evaluate them? Whom do we call on to evaluate them? Who are we going to trust? And it's unclear. It's totally unclear. And the FDA is not going to do this, because they certainly can't scale the number of possible decision systems that might exist. I don't think there's any federal agency that has this in its mandate, and I think it represents a real challenge.
Tony, I see your hand is up.
Yeah. Mark, in your tape conversation, the question was raised about semantic closure, inferential closure, which rule-based systems are at least susceptible to. But, well, at least Karen suggested LLMs not so much. And it strikes me, though, as paradoxical that in that case, we know that it isn't that hard to get a contradiction, a flat contradiction out of an LLM. There have been, plenty of examples being published in which, with suitable dialogue, you can get an LLM to contradict itself. In fact, the first one I remember seeing was posted on an EMEA chat almost exactly a year ago. So what hope of knowledge being encoded in an LLM, if that is the case?
You're pointing out a serious problem. And I think, again, if we were to have some kind of entity that would certify LLMs the way it might certify other decision support aids, I don't know how we would get around the problem of dealing with this kind of complexity, because we're still in a situation where the world is very complicated, where LLMs in particular are not deterministic in how they respond to a given set of inputs. And people want to have confidence in the results of decision aids that they're trusting, particularly when those decision aids are dealing with very complicated and serious situations.
Alan, I see your hand is up, and your hand is up.
Two-handed attempt. I'd like to join the conversation that was stimulated by Richard Scheuman's question. We have a challenge here, right? The top-down system-wide approaches have problems dealing with clinical decision-making tasks. The clinical decision-making task focus has problems in dealing with system-wide implementation and integration. So, Mark Musin, what I think we need is a new taxonomy that will establish a metadata system for characterizing returns from task-oriented clinical decision support tools. In other words, if you have a patient with multiple problems and you run 4 protocols simultaneously, which we have done, and they're all followed, 95, 97% of the time. So they're almost automated. How do you integrate these so that it works to make a clinical care program that's manageable for the patient? Well, what we don't have right now are metadata that characterize the returns from these protocols. In other words, are they urgent? And what do we mean by urgency? Are they life-threatening? Are they important to the patient? Right? In a sepsis patient, losing the tip of your 5th digit on your right hand would be a rather inconsequential issue if we could save the person's life, unless the person happened to be a 26-year-old world-class violinist and she needed that digit. So what I'm trying to point out is that if we focus, in addition to all these other things that are so exciting, on how to characterize the returns from the specific task-oriented clinical decision support tools, we have a chance of getting to an integrated program that would bring the bottom-up task-oriented focus and the top-down IT system focus to converge. And as just an additional comment, what Dr. Scheuermann pointed out clearly is going to need automated returns that are focused on specific clinical tasks, because the database is just too large to expect any human being to be able to deal with it at a level that would be meaningful.
I agree with you completely. And I think ever since MCBK started, there have been conversations in the group about how to create metadata for computable objects. And I think the advent of LLMs makes that conversation even more important. When we first began these conversations, we were talking about rule-based systems and other kinds of decision aids, which require a lot of time and effort to build. Now, as I said earlier, creating a machine learning system or an LLM is almost applying a commodity, and we can do that pretty quickly. And yet, when we're done, we can see performance, which looks pretty impressive on the surface, without really understanding where are the limitations, what is this thing really good for, how do we apply it, how can we have confidence in it. And I think it means that until we have the right way of talking about these kinds of artifacts in a uniform language that we can use as metadata, that we can use to search for artifacts, that we can use to retrieve them and validate them, we're going to be in trouble, because the ability to create thousands of these things now has just increased.
Yes, my argument is that the metadata we need for clinical decision support are not the metadata describing the artifacts, but describing the returns from the artifacts when they're presented with comprehensive and pertinent input data.
Right. We need to describe how is the artifact constructed, what is the content that it operates on, what is the clinical situation in which that content gets brought to bear, and why is it important. I mean, there's a whole range of things, I think.
Mark, I wonder if some of that has to deal with the word trust, which has come up a few times. And have you had any additional thoughts or thinking in terms of trustable LLMs or an AI since the first conversation you had with Dr. Versford?
Oh, it's a great question. I wish I had a good answer for you. I think the problem with LLMs is that we all know that they're not trustable. And if you ask them if they're trustable, they'll just say yes. I the element of hallucination or confabulation is always so high that it's very hard for a third party to be able to assess the trustability of any particular artifact. It's a lot easier with a more traditional computational system, which is more deterministic and which is more traceable. In the case of an LLM, that's really hard, and I still don't have a good answer for that one, but it's absolutely critical. And I think, frankly, one of the things that may be valuable in terms of the kinds of metadata we were just talking about, although it's not going to solve the problem definitively, would be the idea of letting third parties know to what degree does a community trust a particular solution. It's not obviously the answer that we all would like, but at least gives us some measure of faith in a particular artifact.
And maybe along those lines of metadata, I just want to give a quick heads up and shout out to the work being done by our Standards and Infrastructure Working Group led by Jamie McCusker and Rachel Richardson in terms of the CBKMM, which they will present tomorrow. So be on the lookout for the great work that they've been doing there, and look forward to seeing the progress that's been made. Alan, do you have is it a quick comment or question? We have 1 minute left.
Yes, a quick comment about trust, the glue that holds social networks together, right? A lot of it depends not on the artifact, but on the clinician culture. As long as clinicians still link themselves through the Hippocratic model to the temple of Asclepius and divine intervention, it's going to be hard to get any artifact that can trump personal judgment and personal belief. Remember, our first closed loop controller, at least the one I'm aware of, were published in the 1950s. There was closed loop control in Boston of iron lungs, and closed loop control at Columbia as well, and closed loop control of anesthesia using EEGs. And then shortly thereafter, within the next 15 years or 20 years, well, no, a little longer, I misspoke. But thereafter, Lou Shepard and his colleagues at the University of Alabama used automated computer systems to control blood product placement in postoperative cardiothoracic surgery patients. They did 800 patients in sequence. I mean, and I could go on and on about the valid and validated results from clinical trials. And yet the medical community has been resistant to accept any of that stuff. And that's, of course, a complicated issue, but I think part of it is belief systems, which are a real problem for our species.
Well, with that, I think it's been a fantastic discussion and a very vibrant chat. I made note of some of the great comments from Danny McMay, Hongfang Liu, and others who have talked about various aspects of not only the evolving real-world applicability and use of LLMs and what may come, but also the point about LLMs being acceptable by many because of they're aimed to please the end user. And so what is that human factor aspect in LLMs, which we haven't gotten into, but this has been a very enlightening conversation. And I think, Christy, this has been something that we've struck something that's a hot topic that we definitely might be something for this chapter to continue in a future meeting.
Yes, definitely. Definitely so. So this is definitely the topic du jour. It seems like AI and large language models, benefits, doubts, everything is showing up in meetings everywhere these days. And so I look forward to exploring that with this community. As a reminder, we're going to, first of all, I want to thank Dr. Musin for this enlightening conversation and really, I think, seeding this area for us for future contemplation. It's definitely an investment in future conversations. So thank you so much, Mark. And I also want to remind everyone, we're going to be taking a 15-minute break, and then we will be returning at 3:15 p.m. Eastern for the poster and demo presentation. So thank you, everyone, for the terrific discussion and the outstanding presentations we've had so far today. And we'll look forward to seeing you at a quarter after. Thank you. Bye. All right, everybody. Thank you. We are easing back from the break. So welcome back, everyone. We are going to be transitioning over to the posters and demonstration sessions. As you know, and as we mentioned earlier, all of the abstracts are available on the MCBK website. And a link will be put in the chat or has been put in the chat. Let's see. A link will be headed into the chat and in your emails. And we'll open up 2 different rooms, and then you can choose which one to enter to engage with the posters. And then after 45 minutes, we'll all be brought back into the main room, and then you can choose to enter one of 3 demo rooms. So we're going to do, first of all, 2 rooms of demo, or two rooms of posters, and then 3 rooms of demos. And just to get the ball rolling, there are 2 different poster sessions. On topic, it seems like. We do have this AI and machine learning as one of the poster sessions. So that is one option. And then the other option is data and evidence. And I think we've seen how all of these things are essential to work together, but we're looking forward to really being able to focus in on each of these 2 topics in the poster sessions. So I'd like to thank Noor is adding the links for these. The abstracts are in the chat, and we'll have the opportunity to select the room here in a moment. So again, AI and machine learning, and then the second one is data and evidence.
Yep, just give us a moment to get these rooms live for you all to select.
Wonderful, thank you.
Thank you all. We're just regathering back from our rooms. Hopefully you enjoyed your poster session as much as I enjoyed ours. It was a fantastic group, and I want to thank all who have put in the time and the effort to put together such great posters that lay out the fantastic work that you that all have underway, and for taking this time to share and engage in conversation about that work, and look for ways in which we might be able to make some connections through this community in order to support one another and mobilizing computable biomedical knowledge. The next portion of our meeting is to look at participate in and view technical demonstrations. We've broken out these demonstrations into 3 rooms, which you can see on screen here. And like the poster session, you have the opportunity to move in and out of the different rooms. So if there's one one demonstration in one room and you want to move to another, you're able to do so. I think Janelle and Noor, are there any other logistics that folks should know about prior to going into the technical demonstrations?
No, I think we're just waiting on Janelle to publish the 3 breakout rooms. It's just room 1, room 2, room 3. No fancy names. And looks like they're open.
Fantastic. So we invite you to join a breakout room at the bottom of your Zoom screen. You should see a pop-up window. You click on that, and there should be 3 rooms. Those rooms yes, okay, great. And I see people are already starting to join. So we will gather here back in 45 minutes. Enjoy your demonstration. And we are back. Christy, I see that you were with us. I trust that the day's events, the posters, the demos, the speakers. I don't know. Are you as impressed as I am?
I
How awesome this first day has been?
Yes, I am. And I think one of the exciting things is that, uh, here we are coming together. This is the inaugural meeting. And the idea that we're going to just keep growing and improving year over year is, I'm already starting to think about what this might look like next year. But fortunately, we don't have to wait until next year for more of these kinds of conversations because we have tomorrow's program ahead. So that's very exciting. So Josh, what are some of the things that you've observed today that you'd like to share with the group?
Yeah, that's a great question. And I'm still processing so much information, but I mean, going from beginning to end, just very quickly, I was impressed by what our colleagues in the UK are doing, and was making some notes in terms of thinking about what we might be able to emulate or learn from them, given the momentum it seems like they've built there, and what can we do to further build a momentum on our end in terms of CBK. I think that the presentations from our plenary speakers, Richard Scheuerman and Dr. Richard Sherman, Dr. Mark Musin in this conversation really laid the groundwork for some areas for future thinking around LLMs and AI. I think publications of the translation of data into into knowledge sets and ways in which that knowledge is published. And so I think there's going to be some conversations tomorrow on that very topic. The presentations and the posters, I was just very impressed in terms of questions and work that's being done around data representation for knowledge management, implementation challenges, and how those challenges are being addressed by various stakeholders. And I was very interested to see real world applications of how knowledge is being integrated into clinical decision support platforms and interventions. I'll stop there. It was just smorgasbord for me.
Right, absolutely. And I think, one of the things that really stood out to me, I mean, of course, we were just so honored and privileged to get to hear from esteemed colleagues who presented the plenary talks and gave their perspectives. But I do want to highlight the demonstration sessions and the posters were really very inspiring because it gave us a chance to be able to see the kinds of things that people are working on, what is catching their eye, what those investments in their time and energy are. And I think it really inspired me to think about what this might look like in the, in future iterations as we begin to see these things show up in other talks, in the literature, and then next year at the conference. So again, just to reiterate, our great thanks to everybody who participated in the program today, whatever your participation was. And also to give a shout out to the really robust conversation in the chat too. I think this kind of meeting can only be as beneficial and as inspiring as it is when we get a chance to really kind of facilitate that back and forth discussion, which can be hard to do in virtual settings, but I think you all proved that statement wrong today. It was really wonderful. So thank you very much.
You're here. Well said, Christy, for sure. I think also maybe before we getting into tomorrow's events, just another big thanks to the team, MCBK team, Noor, Janelle, for all of you done in supporting this first day of the MCBK NA meeting. Thanks to Chuck for your guidance and leadership and making this all happen. Thanks to our program committee for bringing this to fruition. And thanks to you all for a really robust conversation, as Christy rightfully laid out in the chat, and engaging questions and answers during our sessions. Thank you all. I don't know what's next on our slide deck, but is it the next tomorrow's events? If not, kind of Christy and I can probably relay what who those speakers are and what we have entailed for day 2.
Yeah, I can share. Hang on. Let's see if this shows up on time. Yes, tomorrow's presentation. Oh.
Fantastic. So we're really fortunate to have a keynote from Dr. Aaron Holvey tomorrow, providing Pecori's perspective on mobilizing computable knowledge, biomedical knowledge, particularly around patient-centered comparative effectiveness research. We'll have a discussion with Dr. Ganesh Koru around publishing CBK. We'll hear from Doctors McCusker and Richardson around the work that they have been leading on a CBK metadata model. And we'll hear from, we'll also have a very quick interactive session around MCBK North America governance. And then kind of including or extending that interactivity, we'll be going into what the work groups have been doing over the past year, and laying out discussions with you all at the work group level, what work groups can be doing currently and in the forthcoming year to be more engaging, be more active, and contribute to a growing and vibrant MCBK community. Christy, did I hit all the high points, or is there anything you'd like to add?
I think you did, and I'm looking forward to thinking about the program as it's happening tomorrow and kind of wrapping things up, because I think we're going to continue to see other bright, shining examples of great work in this area, and especially collaborative initiatives, and looking at the work of the work groups and other opportunities to think about how we move forward together as a community. So thank you so much, Josh, and thank you everyone who was able to join today. We're looking forward not only to tomorrow's conversation, but to the ongoing discussion and efforts in order to make some of these thoughts a reality and everyday practice for us. So thanks for being such an inspiring community, and we're looking forward to tomorrow's events. All right, thank you very much, everyone.
Thank you.
See you tomorrow.
See you tomorrow.
Bye.
Bye-bye.
Thank you both.